The working procedure of a steam sterilizer, autoclave, follows a specific cycle to ensure the sterilization of equipment, materials, or products using saturated steam under pressure. Here's a general outline of the process. Preparation, loading. Materials, instruments, media, or other equipment are prepared and placed inside the sterilizer. Items are packed in such a way that steam can easily penetrate all surfaces. Seal the door. The door of the autoclave is securely closed, ensuring a proper seal to prevent steam leakage. Cycle initiation. Air removal. The cycle begins with the removal of air from the chamber, as trapped air can interfere with effective steam sterilization. There are different methods for air removal. Gravity displacement. Steam pushes the air out of the chamber through a vent. Pre-vacuum. A vacuum pump removes air from the chamber before steam is introduced. Steam flush pressure pulse, SFPP, alternates between steam injections and vacuum pulses. Sterilization. Steam injection. Steam is introduced into the chamber. The temperature rises to the set sterilization temperature, typically between 121 degrees Celsius to 134 degrees Celsius, 250 degrees Fahrenheit to 273 degrees Fahrenheit. Pressure buildup. The steam creates pressure inside the chamber, usually around 15 to 30 psi depending on the sterilization cycle requirements. Holding time. The sterilizer maintains the set temperature and pressure for a specific period, usually 15 to 30 minutes, ensuring complete microbial destruction, including spores. Select the sterilization cycle. Choose the appropriate cycle. Depending on the type of materials being sterilized, select the correct sterilization cycle. Common options include 121 degrees Celsius, 250 degrees Fahrenheit, at 15 psi for 15 to 30 minutes for standard sterilization of most materials. 134 degrees Celsius, 273 degrees Fahrenheit, at 30 psi for shorter cycles for heat-resistant materials. Some sterilizers allow you to customize cycles based on temperature, time, and drying phases. Sterilization time depends on the load type, volume, and temperature settings. Exhaust and drying. Steam exhaust. After the sterilization hold time, the steam is gradually vented from the chamber, and pressure is released. Drying phase, optional. For wrapped items or certain loads, a vacuum or heated drying phase is often included to ensure the sterilized items are completely dry, preventing recontamination. Cooling and unloading. Cooling. Once the sterilization cycle is complete and the pressure has returned to ambient levels, the chamber and its contents cool down. Unloading. After cooling, the door is opened, and the sterilized items are removed. Care must be taken to handle items with sterile techniques to avoid contamination. Cycle validation and record keeping. Cycle validation. Indicators such as biological, chemical, or physical indicators are often used to ensure the sterilization parameters, time, temperature, pressure, have been met. Record keeping. Autoclaves usually generate printouts or digital records of the cycle, including temperature, pressure, and time data, which must be reviewed and archived for regulatory compliance. This procedure ensures that materials are properly sterilized for use in sterile manufacturing, surgical procedures, and laboratory environments, meeting CGMP requirements. To work with a steam sterilizer, autoclave, effectively, you need to follow a structured process to ensure safety, efficacy, and regulatory compliance.